Hello, my lovely and wonderful humans. So as I promised, I am going to be working through this assignment with you guys. Now, this is an assignment that we should have received Thursday. No, we received this on Wednesday. So Wednesday would have been January 12th. Anyways, so you guys got this little booklet here. Okay, 3-page booklet. Don't forget that if you guys color this in, I will give you an extra 5 points, okay? And really, there's not much to color in, so you might as well. Why not? Everybody could use a little extra credit, right? Okay, but since you guys are having such a hard time, like actually reading all of my answers and stuff, while we were going through this in class, what I ended up doing was printing off a bigger version of this. So I'm gonna go through this with you guys on this larger assignment here. Just to make sure that you guys are able to read and see what's actually going on. But do know that this is the same exact thing as that folded assignment that you guys did get, okay? So first things first, right? We got to put a name on here. So my name is Ms. Reyes, okay? You guys put yours. Okay, so first things, we got to get started. So we have an example genetic cross, okay? So we have a heterozygous tall pea plant that is going to be crossed with another heterozygous tall plant. Tall is dominant over short. So we need to calculate the phenotype. And what is the phenotype? That's right. Phenotype is physical trait. And I'm going to write that up here so we don't forget. So phenotype, okay, equals physical traits. Okay, physical traits. Now, remember what I said, that phenotype word starts with a PH and so does physical. So that's how I remember it, at least. I don't know about you guys. Anyways, so back to this, they want us to calculate the phenotype and the genotype, and genotype, right? Genotype is what we would basically call our code or our DNA, okay? And our DNA is basically coded. So we call this our coding. So what is our coding? Okay, so we need to calculate the phenotype and the genotype ratios of these two pea plants, what their babies would look like. All right, so question number 1, which trait / allele is dominant? So as we can see here, tall is dominant. So we're going to go ahead and write tall. And while we're doing this, we're also just going to go ahead and assign it with an allele. And we're going to use the letter T. And because it's dominant, we're using a capital letter of this version. Okay. So as we can see, this next question asks, which is recessive? And we can see here that tall is dominant over short. So that must mean that short must be recessive. And in order for us to write an allele, right? For recessive alleles, we always use the lowercase version of the letter that we're using. It's just how it works. Okay, so from question 1, we need to move on to number 2. And number 2 is going to ask us to write each parent's phenotype, okay? And remember, phenotype means what? Physical traits, okay? Now, I don't know if you guys noticed this, but I am going to be saying a lot of these vocab words often and again and again and again. And the reason is because it is so important that you guys actually remember and understand the two things, because they're big words, right? They're big biology vocab words, but they are incredibly important to understanding what's going on, especially for what we're doing here for the next few weeks, okay? So back to phenotype. Once again, phenotype we know is physical. And I'm just going to make a little note of that here so I just don't forget, right? So it's asking our phenotype. So we're looking for the physical trait. So what was the physical trait of parent number 1? Well, let's go back up here. Let's assign this guy up here as parent number 1. And we'll call this guy, because he comes in second, as parent number 2. So as we know, the phenotype is the physical trait. Well, up here, the physical trait is not heterozygous, but it is tall. So our parent number one's phenotype is going to be tall. Okay. All right, on to parent number two. So if we look at parent number two here, we will see that the phenotype, and once again, the phenotype is the physical trait. The phenotype, again, we have heterozygous, that is not a phenotype, but tall is a phenotype. So we can go ahead and write tall in that spot. Moving on to this next one, genotype. We said that genotype was this coding, right? It's like our DNA. So I'm gonna, once again, go ahead and write it just because it really helps to remember, okay? And so now we're gonna be looking for our coding, right? So up here, we wrote a capital T for the dominant allele, and we wrote a lowercase t for recessive. So we're gonna be using the letter T to definitely make our coding here. But the thing is, our coding is always made up of two alleles. So we need two letters. The thing is, do we really know what two letters we need? So we need to go back up to the problem here. And once again, read through here. So we have heterozygous, we have tall. Okay, so we know that tall referred to the physical trait. So we know now that heterozygous is going to refer to our genotype. And heterozygous, I don't know if you guys remember this, but we have heterozygous, which is going to mean, that's right, opposite or different. Okay, and that just means that our two alleles are going to be either, they're going to be opposite or different. Alternatively, instead of heterozygous, what we can have is called homozygous. All right, we can have homozygous. And that means homo is going to tell us that those alleles are going to be the same. So again, if we go back up here, we see that our P plant is going to be heterozygous. And again, we know that heterozygous means that we have opposite alleles. So if we have these two as our alleles, we know that the two alleles for parent number one are going to be opposite each other. So a capital T and a lowercase t. Okay, on to parent number two. So once again, we look at our problem over here and we see that parent number two is also heterozygous. And heterozygous can literally only mean one thing. And that one thing is that they are definitely going to be opposite. So just like we have over here with parent number one, we're also going to have a capital T, lowercase t, or dominant and recessive allele. Okay. All right, lastly, alleles. So each one of these letters is an allele. So here we see two alleles. Here we see two alleles. Now, this is asking what possible allele parent number one could donate to its babies. So we have two options here. We have capital T and we have lowercase t. And we're just going to write just that. Okay, so instead of having them paired here, because this is asking only which are the alleles, we're going to put them as single split by a comma. And again, because parent number two basically has that same exact genotype, we can do just about the same for the alleles. They both are going to be able to provide either a dominant allele or a recessive allele to their babies. All right, after two comes three. And so this is going to be paired with this box here, but this gives us a little bit of context about what's going on down here. So let's start up here. Number three tells us to fill in the parent alleles on the top and on the left side of the Punnett square. So this right here is one of those Punnett squares, okay? And this guy over here, we're gonna call this one parent number one. And we're going to call this dude over here parent number two. So parent number one has this genotype, a capital T and a lowercase t, as we wrote over here. Now we know that we need to split up their alleles. So we're going to go ahead and put that capital T over here and the lowercase t over here. And again, this could be dominant T or a recessive T. Over here, we see that our parent number two has similar alleles. So we once again want to go ahead and split them up in the box. Okay. All right. So I think we're done with number 3. Now we can move on to number 4. So we're going to fill in each box with the allele from from the top parent and the allele from the left parent. Okay. So back to our little Punnett square here. So basically the way that Punnett squares works is that parents can only donate one allele because babies only have 2 alleles. So a parent is going to donate one and the other parent is going to donate the other. Now, when we do this, we're able to see what types of babies are possible. So what parent number 1 will donate to its baby number 1 here, is going to be that capital T that we see above here. Okay, and same thing for parent number 2, except we're going to drag it from the left over to the right, and we are going to still have another capital T. Okay, so baby number 1 is going to have what we call a homozygous dominant. Okay, let's break that word down. So homozygous, right? We wrote it over here. Homozygous means that we have the same alleles. So as we can see here, our alleles are both capital. Now, dominant, on the other hand, is going to tell us that it is the allele that's always going to show up whenever it's in the presence of a recessive allele. Okay, so we get homozygous dominant for baby number 1. All right, on to baby number 2. So as we can see, parent number 1 is going to donate a recessive allele. Now, the thing with this that we need to remember is that we don't quite know what parent number 2 is going to donate. And because we always put the recessive allele at the end, we need to make sure that we're putting that T here over on the side. Because once again, we don't know yet what parent number 2 is going to give to baby. So we're going to end with that first allele, okay? Now we can move on to parent number 2 and see that parent number 2 is going to give baby number 2 a dominant allele. So now we have what is called a heterozygous baby. And our heterozygous baby just means that he has opposite alleles. Okay, so we have a dominant and we have a recessive allele. All right, on to baby number 3 over here in this box. So we look at what parent number 1 is going to donate to baby number 3, and we see that it is a capital T. And because we know we always are going to put the dominant allele first, we are going to make sure that we put that capital T first. Okay, then we can move on to parent number 2. And with parent number 2, we see that we have a recessive allele. So I'm going to go ahead and put that in for baby number 3. Now, just like baby number 2, baby number 3 has heterozygous alleles, right? Those alleles are opposite or different. So baby number 2 and baby number 3 are heterozygous. And lastly, we have baby number 4 right here in this very last box that we have yet to fill in. So let's look at what parent number 1 is going to give to baby number 4. Parent number 1 is going to give that recessive allele. And once again, because we don't know yet what parent number 2 is going to give baby, we're going to put that recessive allele towards the end. Then we can look at parent number 2 and see that parent number 2 only has a recessive allele to donate. So now what we have are 2 of the same alleles, and we know that we call that homozygous. But the kinds of alleles that we have here are called recessive. So we would call this homozygous recessive. And that distinguishes the difference between homozygous dominant and homozygous recessive, because they are very different both in genotype and in phenotype. So both in coding and in physical trait. Alrighty, so I think we're good to move on to question number 5 here. So let me just bump down my camera so you guys can go ahead and see that. All right, so for number 5, it asks us to write the genotype ratio. And once again, genotype, oh, that means code. So we need to write the coding ratio of the offspring by counting how many of each genotype. Once again, genotype means code. Writing each one of the code in the Punnett squares offspring boxes, or the babies boxes, right? So we need to figure out what types of babies we could possibly have. So the first type of baby we could possibly have is going to be that homozygous dominant baby, right? So we have a homozygous dominant baby right here. And I'm just going to fill that in to make sure that you guys can see exactly what I am talking about. And now I'm just stalling so that I could color it in and still kind of fulfill my OCD low-key vibes. All right, anyways. Okay, so as we can see here, we are going to have a homozygous dominant baby. Okay. The next type of baby we could potentially see is this heterozygous baby. Okay, and heterozygous means, once again, that we have opposite or different alleles. And we have a homozygous baby, or sorry, we have a heterozygous baby here. So I'm going to go ahead and highlight this baby in green and once again stall because, you know, now I have to color in 2 boxes, right? Because we don't just have baby number 2 as a heterozygote. We also have baby number 3 as a heterozygote. Sorry, guys. Like I said, low-key OCD. All righty. That's about it. Yeah, that'll do. Okay, so we have our heterozygote here. So we need to go ahead and add that into our ratio over here. Now, these little colons here, okay, those little dots, they're going to separate the different types of alleles. So I just went ahead and made sure that you guys could see them. So again, we have heterozygous alleles here. So I'm going to make sure that I write my heterozygous alleles over there. And the last and final type of allele that we could potentially see with these babies, with the parents of these babies, is going to be that homozygous recessive. And once again, homozygous is going to mean same, and recessive just refers to that trait that typically we don't see, especially if it's in the presence of a dominant allele. Okay, so that means we have 2 lowercase t's, and we're going to go ahead and add that to the potential genotypes our babies could have. So now it asks us to count or figure out what the ratio would be. So how many of these homozygous dominant babies, how many of these heterozygous babies, and how many of these homozygous recessive babies could we potentially have if we were to have 4 babies? Well, as you can see here, we have only 1 baby that is going to be homozygous dominant. So I can go ahead and put a 1 in there. I have 2 babies that could potentially have this type of genotype coding. And so I'm going to go ahead and write a 2 in that spot. And then lastly, we can see that we only have 1 baby that could potentially have a homozygous recessive genotype. So I'm going to go ahead and add a 1. So we would call this a 1:2:1 ratio. And that just means that for every 2 heterozygous babies, we will have 1 that is homozygous dominant and 1 that is homozygous recessive. Just kind of tells us what the frequency or the likelihood that something or a certain genotype will appear, okay? All right, and we go down to number 6. So now it asks us to write the phenotype ratio. So a phenotype, as we know, is going to be that physical trait. So what are the potential physical traits that we could see? So now we need to look at these. So over here, we see a homozygous dominant allele. We know for sure, all we're going to see is whatever the dominant trait is. And as we identify the dominant trait over here, we know that those babies are for sure going to be tall. So we can go ahead and write that over here. So let's look at our next type of babies, which is these heterozygous babies. Now, what we know about heterozygous babies is that we have opposite or different alleles, and we will always have a dominant and a recessive allele. Now, literally the definition of dominant is that dominant will always appear when in the presence of a recessive allele. So we know that this baby will always show the dominant trait. And again, we know that the dominant trait is going to be that tall phenotype. So we can include these babies under the tall category. So we have 1 potential baby as a homozygous dominant, and we have 2 potential babies. So in total, we will have 3 tall babies, okay? And the other option for the phenotype is that homozygous recessive allele is going to code for. And that trait, if we look at the recessive trait, we are going to see a short plant. So I'm going to go ahead and add that to that spot right there. Now, there's only one baby that could potentially come from these parents in the potential for 4 babies. We will only see 1 baby for every 3 tall babies. Okay. And the reason, because again, is whenever we're paired with a dominant allele, we will only ever see the dominant allele. Even though that recessive gene is still there, we will always only ever see the dominant allele. All right, and on to this last box over here for the first page, okay? Remember, there's 3 pages here. I'm so sorry, but stick with me, okay? We're gonna get through this. Trust me, Miss Reyes wants to go home and feed her puppy. Anyways, so we have Punnett squares are named after the man who invented them. They are what we call a tool, and it's gonna help us to figure out all of the possible combinations of the alleles that an offspring or a baby could potentially have. The second thing that it's going to help us to figure out would be the probability that each allele combination is going to appear in the babies or in the offspring. Now, my question to you guys is, what the heck is probability? Probability. What does that mean? So probability just refers to the likelihood that something will happen. Okay, so probability is the likelihood that something will happen. So if we're using this as our example, we will see that, again, for every 3 tall babies, we will only ever see 1 short baby. If we were to put that into percent, right, we have 4 total babies, which means that 75% of our tall would be tall babies and only 25% would be short babies, okay? Alrighty, so let's go ahead and move on to the second page. We are getting through it, guys. I'm so proud of you. Okay, so now we're gonna be looking at some zebrafish here. So we have clues from relatives. A young striped zebrafish, so our zebrafish in question is striped, has a striped father and a spotted mother. Stripes is going to be dominant over spots, okay? So our first question here is, what is the genotype? And once again, what's a genotype? That's right, it's the coding. So the genotype or the coding of the young striped zebrafish. So this dude over here. Well, first we need to figure out what kind of coding dad is gonna have. So we have dad up here. Now dad, it says for dad that dad is striped. So we definitely know that because stripes is dominant, we know for sure that dad has a dominant allele. And instead of using S, we're gonna use F for fish, okay? Because it's kind of hard to tell the difference between a capital S and a lowercase s. But if we use an F, it's way easier to see. Anyways, as I was saying, dad definitely is going to have a dominant allele because we see the dominant allele. So he's at least gonna have 1. Now, we don't exactly know what his second one is quite just yet, but if we take a look at the mom, okay, the mom, on the other hand, is spotted, okay? And because we know that stripes is gonna be dominant, we can assume that spotted is going to be recessive. So we definitely know what our mom's genotype is going to be here. And because she's showing a recessive trait, and a recessive trait will only ever show if there is no dominant trait present, we know that mom's coding is definitely for sure going to be 2 lowercase f's or 2 recessive f's or 2 recessive alleles, okay? Which means that baby, okay, baby is for sure going to have a capital F. And since mom literally only has recessive alleles to give to that baby, baby could literally only ever have a heterozygous genotype. Okay, so a capital F and a lowercase f genotype or coding. All right, so moving on to here. Because the young fish has stripes and its mother was homozygous for the recessive trait spots, you can tell that the young fish is a heterozygote, right? Heterozygous means that we have opposite or different. So this fish is going to be a heterozygote. All right. Because it must have inherited which allele from its mom? Again, mom could only potentially give that recessive allele, because that is literally the only kind of allele that she has to give. Alrighty, moving on to hybrid. So we have an albino male mouse who is going to be mated with a hybrid brown-haired female mouse. Okay, so what do we know about albinism for sure? That's right, albinism is a recessive trait. We will only ever see albinism occur when it is in a recessive pairing, so a homozygous recessive pairing. So we know for sure, and if we're using B for brown, we know for sure that this mouse has albinism. We know that its genotype is going to be a lowercase b and lowercase b, or recessive allele and recessive allele. Now, hybrid. Let's talk about what hybrid means. So if you think about a hybrid car, what is a hybrid car? Well, a hybrid car runs on both gas and on electricity. So are those two things the same or are they different? That's right. Gas and electricity are 2 different types of energy. So what do we know that with genotypes is different as opposed to the same? Exactly. So hybrid here is just going to mean that it is a heterozygous, or it has a heterozygous allele. Heterozygous allele pairing, okay? So in this example, because the heterozygote will always show the dominant traits, because the heterozygote will only ever show the dominant trait, brown hair, we know, is going to be the dominant traits. All right, so we sidetracked here a little, but let's go back to the main question here. So now it's asking, what is the genotype of the female parent? Well, let's take a look at that female parent here. It says she's a hybrid. And again, because we know that hybrid means heterozygous, we for sure know that her allele pairing could only be those that are opposite or different from each other, which means that she's absolutely going to have a capital B and a lowercase b. Alrighty, on to true breeding here. Okay, so with true breeding, brown-bodied male fly is going to mate with a white-bodied female fly. The brown body allele is dominant over the white body allele, okay? And once again, we're using B for brown and lowercase b for white. So it's asking us, what is the genotype of the male parent fly? Well, we definitely know that white-bodied is going to be the recessive allele, because it says that the brown body is going to be dominant. So we know that this female is definitely going to have a homozygous recessive pairing. And because we're using B, we're going to use 2 lowercase b's. So our mom is going to have a homozygous recessive allele pairing. Our father, on the other hand, okay, he is brown bodied. So we know for sure, at least one of his alleles is going to be a dominant allele. So he's definitely going to have at least one capital B. Now that doesn't necessarily mean that we know what the second allele is going to be. But what if I tell you that true breeding means that we are going to breed to homozygous individuals? All right. So we will always breed two homozygous individuals. So as we can see here, we know that this mom is a homozygous recessive individual. So because our male is brown, we know that he is homozygous, we know that he is going to have two capital B alleles or two dominant alleles. So when it asks what is the genotype, again, coding of the male parent fly, we can say that it's going to be a homozygous dominant pairing or two capital B's. Alrighty, and on to the home stretch here, okay? So we need to look at dominant or recessive. So we have a heterozygous round-seeded pea plant that is going to be crossed with a wrinkle-seeded pea plant. So the question asks which allele is dominant. Well, with heterozygous, right? Whenever we have a heterozygous pairing, we know for sure that heterozygous means opposite or different. So we know that the alleles are going to be opposite one another. So for this, we're going to use R. And because it's heterozygous, we know we're going to have a capital R and a lowercase r, or a dominant allele and a recessive allele. So that's parent number one there, the heterozygous parent. Now, because we only ever will see the dominant allele whenever it's present with a recessive allele, we can assume that round is going to be dominant. A round seed is going to be the dominant trait here. And because we're using R, we know that it's going to be coded with a capital R. Now, if we go back to the wrinkle-seeded plant, this is clearly the recessive trait. And if we are seeing the recessive trait, we know that it has to be a homozygous recessive pairing. So we know that this plant definitely has homozygous recessive alleles. So just to sum that up, in order to figure out which allele is dominant, all we have to do is look at the phenotype. And what's phenotype? That's right, it's the physical trait. All we have to do is look at the physical trait of a heterozygous individual in the problem. The heterozygous individual is always going to show the dominant trait. Will always show the dominant trait, not the recessive. Again, that's literally the definition of dominant and recessive. Dominant will always be the one that appears whenever it is in the presence of the other type of allele. Alrighty, guys, we are so close to being done. I promise this is the last page. I'm so proud of you guys for making it this far, but let's just get it done, okay? All right, so a little bit of background about goldfish. So in goldfish, there's a trait that is caused by a dominant allele in a developmental gene called bubble eye. So dominant goes in here. Okay, so that dominant gene is that bubble eye. So we know that bubble-eyed goldfish are going to have a large bubble-like sac near each eye, and they're also going to lack a dorsal fin. So here we see two goldfish. Here we see good old bubble eye fish, and he's got a little bubble eye, but he's also lacking the dorsal fin that our normal eye dude over here has. So just a quick, easy way to see the differences between these two fish. Now, or normal eyes, lowercase b, is going to be what to bubble eyes or dominant B? It's going to be recessive. And there's a couple of ways we know that, right? So first off, like probably the easiest and quickest way to look at it is that this has a lowercase letter. And we know that the lowercase letter always refers to the recessive allele. It also tells us over here that bubble eye is going to be dominant. So the only other option is recessive, and therefore normal eyes must be recessive. Okie doke, let's move on to this next section. Your friend is going to give you 2 new goldfish. Oh, what a good friend. You want to know what their genotype is. What's the genotype again? That's right, the code. So we need to figure out the coding. So over here, we have our first fish. Now, this is a normal fish. We don't see a bubble eye, but we do see a dorsal fin. So this is our normal fish. And we know that normal eyes is recessive. So if we see that normal trait being expressed, we know for sure the allele pairing is going to be exactly homozygous recessive. So little b, little b. All right, let's look at our fish number 2 over here. So this dude, he does have a bubble sac. So what's his genotype? It could be one of 2 things. So because we know he's showing the dominant trait, we know for sure that he's got a dominant allele. But he could also be expressing this if he were heterozygous or if he were homozygous dominant, right? So he has 2 different options. He could be either homozygous dominant or he could be heterozygous, and he would still appear with a bubble eye. Ooh, guys, we're almost there. This is the last section. So we want to figure out for sure what this dude's genotype is. So we're going to do what is called a test cross, and scientists use test cross to see if an individual organism is going to be heterozygous or, and what's the other option here? That's right, homozygous dominant. Okay, so we want to see whether he's going to be heterozygous or homozygous dominant for a particular trait. So the test cross means that we're going to go ahead and see what babies these 2 are going to make. And we're going to, in order to see what kind of babies they're going to make, we're going to cross them with a genotype that we do know. And that is going to be the genotype of a homozygous recessive individual. We are only going to see that with a homozygous recessive individual. Okay, so now we have to do a little bit of work on the side here in order to figure this out. So I'm going to go ahead and grab a little Post-it note because we need to do what that tool, that handy dandy tool to figure out what our babies are going to look like, right? And that little tool is called a Punnett square. So I'm going to pull out a Post-it note here so you guys can see for sure what's going on. All right, so first off, we need to test the heterozygous pairing here. So we're going to get our heterozygous fish over here, and we're going to start with him. So we got a heterozygous, we've got a dominant paired with a recessive, and we're going to cross that with a homozygous recessive individual. So our lowercase b. So this is the Punnett square we need to run. Now, the Punnett square that we're going to use is made with what is basically a tic-tac-toe box. Yeah, tic-tac-toe back in our lives and in chemistry, no less. So what we do here now is we need to go ahead and separate the alleles for their parents. So we're going to call this parent number 1, and parent number 1 we're going to put here at the top. So again, we need to split up their alleles. So we're going to have capital B and we're going to have lowercase b. Then we're going to go to parent number 2, and parent number 2 is going to go down here to the left, and we're going to split up those alleles. So lowercase b. Now, parent number 1, we need to see what parent number 1 is going to be able to donate to baby number 1. And as we see here, it's that capital B. So we're going to just drop that down into baby number 1's genotype. Parent number 2 is only going to have that lowercase b to donate. So we're going to go ahead and put that lowercase b there. If we move on to baby number 2, what we're going to see is that parent number 1 is going to donate that lowercase b. And again, we need to write that lowercase b at the end because we don't know what's coming from parent number 2 yet. But if we look at parent number 2, we see that parent number 2 has that recessive allele. So now we have a homozygous recessive baby. Okay, on to baby number 3 down here. Now, parent number 1 has that capital B to donate, and so that's going to go in the front because dominant always goes first. And as we see over here with parent number 2 has a lowercase b to give. And lastly, we have baby number 4. So if we look at what parent number 1 has for baby number 4, we see that it's a lowercase b. And again, I put that towards the end because I don't know what parent number 2 is going to donate quite just yet. But if we do look at parent number 2, we see that parent number 2 is going to donate that recessive allele. All right, so what kind of genotypes do we see here? We see, let's see, we see a heterozygous pairing, and we also see a homozygous pairing, right? So what would this guy look like? What would this guy look like? So again, we always will see the dominant trait whenever it's in presence of a recessive trait. And because we know the dominant trait, right? What's the dominant trait? Oh, the bubble eye. We know that this little guy over here is going to have a bubble eye. And I'm just going to write bubble because I'm running out of space. Okay. Now the recessive allele, right, is going to be that normal eyes. So we know that this baby is going to have normal eyes. Now, the second part of this is calculating the frequency, or how frequently we are going to see which type of baby. So if we count how many babies out of this 4, we will see that for every 4 babies that these guys have, about 2 or half of them will have that heterozygous pairing, and half would be 50%. Now, if we look at our homozygous recessive babies, we see that there would be 2 babies for every 4 babies born, which means that, again, we will have a 50% frequency of seeing either one of those. Okay? All right, so that's not the only thing we have here to test. Now we need to test that homozygous dominant. So I'm going to go ahead and get another sticky note. This one's orange. Okay, so I'm going to get another sticky note, and now we need to test that homozygous dominant pairing. So here we have the 2 capital B's, and we're going to pair that once again with our homozygous recessive individual. Okay. Alrighty, so now that we've got that set up, we need to set up that good old Punnett square. So I'm going to go ahead and draw my tic-tac-toe box. Okay. And once again, we need to split up those alleles just like we have over here. So I'm going to put a capital B here, and then for parent number 2, a lowercase b and lowercase b. So if you guys go ahead and do this and fill it out, right, we'll see that parent number 1 gives to baby number 1 a capital B, and parent number 2 is going to give baby number 1 a lowercase b. So we have that heterozygous trait there. Now, if we look at baby number 2, parent number 1 has that capital B to give baby number 2, and parent number 2 has that lowercase b. Move on to baby number 3. Parent number 1 is going to give a capital B. Parent number 2 is going to give a lowercase b. And if we look finally at baby number 4, we're going to see that parent number 1 has that capital B, and parent number 2 has that lowercase b. So the genotypes that we see here are just that heterozygous trait, which is that capital B, lowercase b. And from over here, we see that that's a bubble eye. And we also know that because we will always see the dominant trait, which is that bubble eye. Okay, so we will see a bubble eye. That's the phenotype. This is the genotype. Now, how often are we going to see that genotype with this type of pairing, with this type of parent pairing? We will always see 100% of the babies with this genotype and phenotype. Okay. All right. So now that we know that, let me put these to the side here. Now we need to look at the offspring here. So what types of offspring do we have? Let's start off here. So I see 1, 2 fish that have bubble eyes and 1, 2, 3 that have normal eyes. Now it's definitely not going to be our 100%. And you know, it's not 5, it's not 4 fish here like we did over here. It is 5. But we know because it is impossible to see any normal eyed babies, and possibility number 2 only has bubble eye babies, we know that this pairing is going to be most likely for possibility number 2. So that homozygous dominant crossed with that homozygous recessive parent, okay? Which means that this could only be this pairing, or the heterozygous paired with the homozygous recessive. All righty, and I will go ahead and get rid of those sticky notes. So that's all I got for you today, okay? Make sure you get this filled out. If you have any questions, you can email me or just ask me in person on Tuesday. All right, I am going to finally get out of here. I need to get my guinea pigs home and I need to go feed my puppies. So I hope you guys have a great weekend. Please enjoy yourselves, but also please, please, please be safe. All right, you guys, bye.